I didn't even say anything. Talk about power here, right? How's everybody doing today? It's Friday. Everybody has a smile on their face. This is good. If not, you should have a smile on your face. It's good. The rain isn't supposed to start until tomorrow. And then it's supposed to come pouring down. So you have another day to look forward to, not raining. That's good. Exam a week from today, so I don't want to take the smile off your face, but that is a fact of life coming up. I have decided I'm going to cover material through Wednesday. So I'm going to go all the way through Wednesday on this exam. So that will keep us busy. And hopefully that won't be too big of a problem for people. Last time I talked about topoisomers, and I talked about topoisomerase. So a topoisomer are things that differ in that sort of twisting configuration from one molecule to another. So two molecules can be topoisomers if they have different writhings, basically. Okay? A topoisomerase is, of course, an enzyme that can interconvert topoisomers. That's basically what a topoisomerase does. Okay? I talked about topoisomerase I, and I talked about topoisomerase II. If you find the linking number in business confusing, come see me. I'll be happy to sit down and explain it to you. It's usually one of those things that's easier to sit down and do than it is to do in a lecture. So if that is the case, please come see me. I'll be happy to meet with you. Oh, by the way, I did finally get your regraded exams back in the office. So if you submitted an exam for regrading, I turned it back in this morning. I apologize it took so long. To be honest, it slipped my mind. So what little mind I have. We're not going to go through the mechanism of Topo 1 and Topo 2 other than at the level I talked about it. So the figures that are in the book I think are rather confusing. We're not going to deal with the mechanism as shown in those figures. So you should just rely on the short descriptions I gave about how the two enzymes operate. There are the topoisomerases turn out to be interesting targets for antibiotics. This is important, as you can imagine. If we inhibit topoisomerases, what we do is we inhibit the ability of the cell to relieve tension in DNA. And as we will see, synthesizing DNA can create some tremendous tension. The rate with which this process occurs is phenomenal. And if you plug up topoisomerases, basically the DNA ends up in a knot and the cell can't replicate and the cell will die. So inhibitors of topoisomerases have fairly nasty effects. One of these is nalidixic acid. Again, you don't need to know the structure, but it is a molecule that inhibits topoisomerase II in E. coli, also known as DNA gyrase. Another inhibitor of topoisomerase II is ciprofloxacin. You hear it described as the antibiotic Cipro, and that is in fact how it works and does its thing. I didn't list an inhibitor of topoisomerase I on here, but I will give you the name of one. It's called camptothecin, C-A-M-P-T-O-T-H-E-C-I-N. Camptothecin inhibits topoisomerase I, and that's C-A-M-P-T-O-T-H-E-C-I-N, camptothecin, inhibitor of topoisomerase I. Now, topoisomerases are critical. And it turns out that most cells have multiple topoisomerases. You see two here. E. coli has several. And some of them have fairly specialized functions, and we won't really go too much into those. But I do want to tell you about one that's kind of interesting. There is a topoisomerase in E. coli that has the name topoisomerase 7. Okay? And topoisomerase 7 has a very simple way, a very simple function. Its function is untangling interlocked circles of DNA. And it turns out that when you go to replicate the E. coli chromosome is circular. And when you go to replicate that, what you end up with at the end of the replication process are two circles that are literally like this. Okay? That's the way they are. Well, you can imagine that when cell division comes time to go, you obviously can't have these guys interlocked because they're going to go to one place or the other. And the topoisomerase 7 in E. coli, what it does is it does one thing. And that's all it does. Okay? That is, it unlocks and puts them back together, the circles as you see here. That means that targeting topoisomerase 7 is, in fact, another antibiotic strategy, because if you can stop the circles from untangling, you stop cells from dividing. Kind of a cool thing. Okay, so topoisomerases are important targets for antibiotics. All right, well, I want to spend a little bit of time talking about DNA polymerases. So, of course, DNA polymerases are enzymes that catalyze the synthesis of DNA. DNA polymerases catalyze the formation of phosphodiester bonds. The bonds that join adjacent nucleotides are called phosphodiester bonds. That means that there's a phosphate on either side, and on either side of it is a ribose. Okay? A phosphate on either side of it is a ribose. The DNA polymerase not only has to make phosphodiester bonds, but it has to put the right nucleotide in during the synthesis process. That right nucleotide has to base pair with the proper nucleotide on the opposite strands. The structure of DNA is such that the strands are what we call anti-parallel, meaning that we have ends to the DNAs. We have a 5' end and a 3' end. The 5' end is the end that's free, and the numbering is according to the ribose ring. So a 5' end to a 3' end means that we have the 5' most nucleotide here, the 3' most nucleotide here. The strands are arranged so that they're what's called anti-parallel. 5 to 3 here, 5 to 3 here. Okay? So the 3' end here is adjacent to the 5' end here, and the 5' end here is adjacent to the 3' end up here. Okay? So that means then that the DNA polymerase has to move along in that direction. DNA polymerase is always, there's very few alwayses in biology, but here's an always for you. DNA polymerases always build phosphodiester bonds in the 5' to 3' direction. There are no exceptions to that. Always build bonds in the 5' to 3' direction. Okay? Well, this is the structure of a DNA polymerase. It's DNA polymerase I from E. coli. And the structure of this enzyme is interesting. It has the shape sort of like a hand. And we can think of this hand as looking something like I have here, where the DNA double helix will fit in the palm right here of my hand. Okay? So this guy, as it goes, replicating DNA, basically goes sliding along it like this. Okay? Now, it turns out that this enzyme, this particular enzyme, DNA polymerase I, doesn't hold on to DNA very well. It doesn't hold on to DNA very well. It moves along, and then it falls off. It moves along, gets back on, moves along, and falls off. Okay? It is very, what we describe as processive. Okay? It goes on, it pops off. It goes on, it pops off. Actually, I back up. I said it the wrong way. It's very progressive. Sorry, I always do that. It progresses from one strand to another. It moves, pops off, comes over here, pops back on. So it progresses from one to another. An enzyme that goes on and stays on is called processive. Okay? It processes the DNA appropriately. We'll see that one of the DNA polymerases in E. coli is very processive. It gets on, and it basically stays on. And I'll show you why that happens. DNA polymerase I is not one of those enzymes. Okay, Gesundheit. All right. The process of replicating DNA involves a variety of factors. And one of the factors is another enzyme besides the DNA polymerase I. Okay? It turns out that DNA polymerases cannot, and here's another always for you, DNA polymerases cannot start replication of a strand where there's not already one there to start with. Okay? Well, what does that mean? Let's imagine I take DNA and I've got a single strand of DNA. I take this single strand and I lay it out here on this table, and it sits there, and I decide to add DNA polymerase to it and 4 deoxyribonucleotides, which should be all that it needs to replicate, And if I do that, the polymerase will sit there, the DNA will sit there, and absolutely nothing will happen. Zero. Okay? So if I had this template up here, and I put DNA polymerase in there with 4 DNTPs, nothing would happen. It would stay single-stranded, the polymerase would sit and twiddle its thumbs, and nothing would go on. Okay? What I just told you was that the polymerase has to have, the DNA polymerase has to have an existing strand that it extends. It has to extend an existing strand. Okay? Well, if it can't make the strand, how does that existing strand get there? And it turns out that it gets there by action of a separate enzyme called primase. Primase is not a DNA polymerase because DNA polymerases can't put that down. Primase is, in fact, an RNA polymerase. It puts down a short piece of RNA, and RNA polymerases are not inhibited this way like DNA polymerases are. The DNA polymerase I, or other DNA polymerases, in fact, almost any DNA polymerase can do this, can go to the end of this now and start replicating. That is, it has an existing end that it can extend. That molecule right there is known as a primer. In this case, it's an RNA molecule. Okay? Well, you can see this is really nice. It's duplex, but then you start thinking, well, wait a minute. What happens with this stuff over here? And we have some considerations about this stuff over here because this is RNA and this is DNA, and we know that the cell ultimately has fully duplex DNA. It doesn't have sections of RNA in there. Okay? Well, in E. coli, this is actually handled relatively easily. E. coli has a circular chromosome. This guy comes all the way back around over here. So if the DNA polymerase gets going, it just copies, 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 and it gets back over here, and all of a sudden, there's that RNA in the way. Okay? That RNA has to be removed in order for the DNA polymerase to fill in the gap with DNA. Because remember, we've got now a DNA primer that's all the way over here that's waiting to come in and do its thing. Okay? E. coli DNA polymerase I has a unique enzymatic activity that the other DNA polymerases in E. coli do not have. It has the ability to chew this out. Okay? The activity has a name. It's called a 5' to 3' exonuclease. So there's a term for you. An exonuclease is an enzyme that starts at the end of a DNA strand, and it chews away nucleotides. So this guy is a 5' to 3' exonuclease. Notice it starts at the 5' end of this guy here, and it starts chewing away the RNA. And behind it, remember this is a DNA polymerase. It also has a DNA polymerase activity. Behind it, it starts putting down DNA. So it gets all the way up to here with DNA. Everybody with me? DNA polymerase I can do that. The other DNA polymerases cannot. DNA polymerase I, for that reason in E. coli, that activity is essential for E. coli to be alive. If you kill the ability of E. coli to remove RNA primers, the E. coli will die. Because you have to eventually have a fully duplex DNA. Now, there's one thing missing here. And the one thing that's missing here is that the DNA polymerase can extend all the way up to here, but it can't join the end of what it made with the beginning of this. There's a little tiny gap. There's one last phosphodiester bond it can't make. That last phosphodiester bond has to be joined together because we want to make a completely intact circle. And that last phosphodiester bond requires action of an enzyme known as DNA ligase. DNA ligase will, in fact, close that gap by making a phosphodiester bond against that piece that has just been made. Okay? Everybody with me? Questions on that? Okay. Well, I've given you a very simple version of the replication of DNA. There's more complexity to it, and yes, I'm going to tell you some more of that complexity. Okay? This shows what we describe as a replication fork. On the last slide, I only showed you the replication of one strand. And I assumed that one strand was circular, and it came all the way around, and we were able to remove the primer. In fact, E. coli has double-stranded DNA, and both strands have to be replicated. Now, this process I'm going to describe to you is not unique to E. coli. Virtually every cell on the face of the earth uses the same general scheme. The names of the enzymes may vary, but the same general scheme carries. Okay? Replication occurs on a chromosomal DNA in what's called a replication fork. You see the fork right here. Okay? What you saw on the last slide was something that started over here with a primer, and it's now bulling its way through and replicating and replicating and replicating and replicating and replicating and replicating and replicating. Okay? That replication has to occur in the 5' to 3' direction, because that was the first rule I gave you. It's absolute in the 5' to 3' direction. Okay? Well, this strand is oriented 5' to 3' this way. It means I can't simultaneously make this strand going this direction, because that would be going in the 3' to 5' direction. Okay? I can't do that. I can't make DNA in the 3' to 5' direction. I've got to make it in the 5' to 3' direction. The only way I can make DNA in the 5' to 3' direction is if, as this strand is being opened up, the DNA polymerase starts making strands back this way. Okay? It turns out they're in pieces. The reason they're in pieces is because DNA polymerase can only work 5' to 3'. Here's the 5' end of a piece. Here's the 3' end of a piece. Here's the 5'. Here's the 3'. Why does it do it in pieces? Well, when this part gets opened up, this piece gets made. But then as the polymerase moves along, a new stretch opens up, and the polymerase says, oh, we start here. And then a new stretch opens up, and it starts here. And you said, but wait, the polymerase can't start a strand. Doesn't primase have to start the strand? Yes. Primase has to come in. So this becomes a very complicated process. It has to be orchestrated very cleverly, because every time one of these little pieces is made, primase has to come in. It has to make a primer. It has to get out of the way. And DNA polymerase has to fill in. Okay? Now, this means that the two strands of DNA, this DNA molecule that I'm describing to you, are made in fundamentally different ways. One piece is made completely intact. It starts, replication proceeds, and everybody's happy. All right? The other one is made in pieces. Those pieces are called Okazaki fragments. They're named for the person who discovered them. And you say, well, these pieces, we had trouble before when the polymerase came along and it came to the end of this RNA primer, which might be this little piece right here. DNA polymerase I has to chew that out. Is DNA ligase needed again? The answer is yes. And you start to see this orchestration gets really involved. You also discovered there's two DNA polymerases involved here. One that I haven't talked about called DNA Polymerase III, which is actually the one that makes most of the DNA. And the second one is DNA Polymerase I that comes in and deals with the primers. And that's all DNA Polymerase I does. All right, so there's a lot of complexity here, a lot of orchestration that has to happen just to make these pieces so they can eventually be joined together. Yes, sir?
DNA polymerase I in E. coli and humans is different in that the bacteria has the ability to knock off the primer and the humans have.
So the human DNA polymerases are much more complicated than are the E. coli polymerases. Humans have an activity that removes primers, yes, okay? But they don't have it built in the same way that E. coli does here. And we don't call them 1, 2, and 3. We call them by different designations. Okay? Yes, sir?
Since each strand is replicated differently, is there a different mutation rate between the strands?
Good question. Is there a different mutation rate between the two strands because they are replicated differently? There are some suggestions there may be a slightly different rate. Okay? It's not gigantic. There are some suggestions there may be slightly different rates in the mutation rate of the two. Okay. Well, there's a lot of stuff going on here, right? And I'm even sparing you some things I'm going to come back and talk about even further. Yes, sir? What signals RNA polymerase to come in and lay down the primer? All right, very good question. It turns out that there's quite a complex that's here. In fact, if you look, there's some videos I've posted on the links on the schedule page. I will encourage you to go and look at those links. You'll get a better idea about all these different proteins that are coming in and doing their thing. What likely happens is there's a protein, for example, called single-strand binding protein. Single-strand binding protein comes along, and whenever these strands get opened up, single-strand binding protein kind of covers and protects them. Single-strand binding protein has some affinity for the primase that may be part of the signal, and the actual structure itself may be the other part of the signal that's necessary. There's not a sequence specificity as such. Okay? All right, so I just gave you another protein, single-strand binding protein. Single-strand binding protein turns out to be a very important protein because you can see we've got some gaps here. We've got some places where we have single strands, and single strands are the last thing that the cell wants to have exposed because all it takes is one little accident and a slice, and the cell now has no way to know how to put things back together. So single-strand binding protein has functions, one of which is to bind to that DNA and protect it. Single-strand binding protein also has some affinity for some of the enzymes involved and may well help direct them to the appropriate places. Okay. Now, this is a very high quality artistic work. This actually shows you one direction of synthesis of a circular chromosome like goes through in E. coli. Okay? This would be if there were no lagging strand synthesis. This is what happens if there is lagging strand synthesis. And you can see that both strands can replicate at the same time if we have lagging strand synthesis. Now, a really remarkable thing is if I go back to, in fact, I'm going to show you right here. If I go to that replication fork, all right, and I look at what's going on there, this shows very nicely many of the players in this process. Okay? I told you that DNA Polymerase III made most of the DNA during the replication process. All right? It turns out that DNA Polymerase III is obviously right here at the fork where the DNA is being made. You say, well, the leading strand is very easy to see. It just moves. What is making the lagging strand? And this will surprise you. It's the same DNA polymerase. DNA Polymerase III literally has to look at the left strand, make it, and then it looks over the right strand and extends a primer. Then it goes back to the left, and then it goes back to the right. There's literally a juggling that the enzyme has to do in order to replicate DNA. All right? There's one DNA polymerase at that fork. Okay? So the leading and the lagging strand are both made by the same DNA Polymerase III. It's a pretty remarkable thing. So DNA Polymerase I only comes into play when the Polymerase III has gotten up next to that DNA next to the primer. Then RNA Polymerase I comes in, chews out the primer, fills it in with DNA, and DNA ligase goes along. Yes, Shannon, you had a question?
So, if it's so processive, how is it letting go of each of these strands and regrounding?
Good question. If it's so processive, how is it able to stay there and not come off? That comes to the next protein. A lot of proteins involved here. Okay? The next protein is something labeled on here called the sliding clamp. It's also called the beta clamp. The beta clamp is a circular protein that binds to the DNA polymerase on one side, and as you see here, it makes a little ring around the duplex DNA on the other side. Because of the sliding clamp, or the beta clamp, I prefer to call it the beta clamp, because of the beta clamp, okay, this polymerase, when it shifts back and forth, doesn't come off. So the beta clamp holds it in place. The reason that DNA Polymerase I is so progressive is because it doesn't bind to the sliding clamp. It has no sliding clamp. It comes in, it goes away, and it falls off. This too would fall off if it didn't stick to the sliding clamp. Yes, ma'am?
So, are there several sliding clamps involved here? So it binds to one on the leading strand, and then DNA pulls through.
One sliding clamp, one polymerase. So there's only one DNA polymerase III that's there, and the DNA polymerase III is having to look back and forth on the leading and lagging strands. Yes?
What do you mean by look? They look back and forth?
Well, it's moving from one strand to the other in terms of what it's doing. Remember, it has to read the bases.
Right.
So it's looking over here, what bases do I put in, puts in the bases, comes back over here, looks, what bases do I put in, and puts in the bases. So it's literally looking at the strands.
So the sliding clamp lets go and regrabs to the different strands?
Say it again.
The sliding clamp is going to
Nope. The sliding clamp stays right there. The enzyme just shifts its position, looking one way versus the other, like you're doing with your head, like this. Okay. If you look in some of the images online, you'll see that actually occurring. And there's at least one image online that I have, and you'll find this confusing, so I'll warn you in advance, that shows it as two enzymes. It's not. It's one enzyme. Okay. The other illustration is correct, but the one that shows two enzymes is not correct. Yes, sir.
So in this illustration, it looks like there are two sliding clamps. Is it just that the sliding clamp is only on the leading strand?
Yeah, that is a little confusing here. This is not what has to happen is that this is illustrating the fact that this is going both ways and that it has to reform a complex in order to make the primer. Okay, that gets into some other areas there. But there's only one polymerase that's at the replication fork. Yes, sir.
How does the polymerase actually get a hold of the nucleotide? Is it just gravity motion, or is there another?
How does the polymerase get a hold of the nucleotide? It turns out that the polymerase has to be assembled on the DNA with the clamp. And this happens at a special sequence. You're actually getting a little ahead of me, but I'll tell you since you asked the question. This happens at a special sequence in the DNA called the origin. How do the nucleotides get there? The nucleotides, that's also a very good question. Let me save that answer for a little bit later. And if you'll remind me if I forget, I will come back to that, okay? Maybe Monday would probably be the day, okay? Okay. All right. Let's see. So, leading, lagging. DNA ligase. Ligase catalyzes the formation of phosphodiester bonds. There's that gap I described to you before. We've got a DNA up here. We've got a DNA back here. That phosphodiester bond has not been made. It takes energy to make the phosphodiester bond. ATP is involved. And once we've done that, we've now closed that little tiny gap that we had between the newly synthesized and the previously synthesized strand. Okay. Let's see. Now, the next thing I want to tell you about, let's go back down to this replication fork again. All right? If we go back to the replication fork, we see some other things on here I haven't described. All right? One is this enzyme up here called helicase. Helicase turns out to have an incredible enzymatic activity and an essential function. All right? One of the things we haven't described is, well, the DNA polymerase wants to replicate a single strand. It wants to look at a strand and replicate it, right? That's what it does. Well, if we look at that, how did that single strand get there? It turns out that cells use an enzyme to untwist the DNA strands. Cells use an enzyme to untwist the DNA strands. The enzyme that they use is called helicase. Okay? Helicase is really good at doing what it does. It takes ATP energy to pull the strands apart. And it literally does, I won't say rotates, but it's like a rotation that it does in unwinding the strands. Now, what it does is remarkable. All right? The process I've been describing to you is pretty amazing in terms of DNA polymerase. DNA polymerase is incredibly accurate, number one. It'll easily do 1 million characters without an error. Easily. Okay? Think of typing 1 million characters, no matter how slowly you type, and not making an error. Okay? Very difficult to imagine doing. Right? Now imagine doing it at the rate of 1,000 characters a second. DNA polymerase is operating at 1,000 characters a second. Yeah, that's pretty amazing. Okay? The entire E. coli chromosome of 6.3 million base pairs can be replicated by 2 DNA polymerases in the grand sum of 20 minutes. Okay? And replicated with incredible accuracy. It does make errors, but don't let me tell you that it doesn't. It does make some errors. But they're on the order of 1 to 1,000,000 to 1 in a billion, depending upon the polymerase that we're talking about. I'll tell you how it keeps from making those errors in a second. For the moment, I want to focus on the helicase. The helicase has to unwind strands faster, or at least as fast, as the DNA polymerase can replicate DNA. Now, this polymerase is making 1,000 nucleotides a second, and it's doing it here, and it's doing it here. It's looking both ways. It's phenomenal what it's doing. Okay? This helicase has to unwind strands to at least keep up with the pace of the DNA polymerase. That means that the helicase has to be rotating and unwinding strands at 6,000 RPM. Now, you engineers who are in here think of an engine running at 6,000 RPM, and you think that's a fairly rapidly moving engine. Imagine that that engine is a little nano machine, smaller than the eye can see, and it's staying ahead of everything and doing that. That is really phenomenal. Okay? The helicase is an amazing enzyme. The helicase, okay, because it's doing this unwinding of the strands, what do you suppose is happening ahead of where the unwinding is occurring? We start seeing tension, right? Remember the tension we talked about? You betcha. Here's how you get tension in replication. This guy is unwinding strands, and ahead of it, if we don't do something about that tension, we're going to make a knot. So further up here, okay, further up here, we have to have a topoisomerase. The topoisomerase that E. coli uses in the replication process is DNA gyrase. Okay? So DNA gyrase has to be up here unwinding things, it has to be untying things or relieving that tension. This guy is unwinding things. Okay, don't confuse, by the way, topoisomerases don't unwind. Right? They simply relieve that tension. Right? They're not unwinding. This guy is doing what I call unwinding. It's peeling the strands apart. Okay? All right, so that's a pretty remarkable process that's going on right here. When I talk about orchestration, there's some pretty amazing orchestration that has to occur in order for DNA polymerase to do its thing. Okay, well, I promised I would tell you how it is that DNA polymerase can be so accurate. All right? So let's spend a minute thinking about the accuracy of DNA polymerase. How am I doing on time? Okay. Almost all cellular DNA polymerases, not absolutely, but most cellular DNA polymerases have another enzymatic activity associated with them. So let's think about our activities first of all. DNA polymerase I, I've described two activities to you. I've described a 5' to 3' activity that makes phosphodiester bonds. That's how we make DNA. We can just simply call it a DNA polymerase activity. I said that the DNA polymerase I also had a 5' to 3' exonuclease activity. That was important for removing those RNA primers. It turns out E. coli DNA polymerase I has a third activity. This is the one that most DNA polymerases have in the cell. This activity is called a 3' to 5' exonuclease activity. And it has nothing to do with RNA. So I'm going to describe to you how that works. Now, E. coli DNA polymerase III also has the 5' to 3' DNA polymerase. It makes phosphodiester bonds in the 5' to 3' direction. It does not have the 5' to 3' exonuclease activity, but it does have the 3' to 5' exonuclease activity I'm getting ready to tell you. Okay? I'll summarize this in a minute. All right. So let's imagine I'm a DNA polymerase that's replicating a strand of DNA. I've got a primer. I've got a long strand up here that I'm in the process of copying. Okay? I'm going to make my DNA in the 5' to 3' direction. So the polymerase comes on here, and it starts putting in. If there's a T, it puts an A. If there's a G, it puts a C. If there's a C, it puts a G. If there's an A, it puts a T. If there's an A, it puts an A. And uh-oh, I just put an A across from an A. Okay? That won't do. That will not do. And polymerase will do that. Doesn't do it very often, but it will do it. You'll mistype, and when you mistype, what do? You hit the backspace key, right? All right? That's exactly what DNA polymerase is doing. At this point, the polymerase will stop and recognize it has made an error, and it will back up. And when it backs up, it starts breaking the very phosphodiester bonds that it put in there in the first place. It'll back up a few nucleotides, each time chewing out one nucleotide at a time, and you'll notice that now it's moving in the 3' to 5' direction, backwards from the direction of the polymerase. This activity, this 3' to 5' exonuclease activity that I've just described to you, is called proofreading. Proofreading lowers the mutation rate of a polymerase by 100 to 1,000-fold, meaning you'll only get 1/100th to 1/1000th the mutations if you have proofreading compared to if you don't. That sort of makes sense. When you type up a paper and you get everything done, you look at it and go, oh my God, I don't know how to spell my name. Go back and you change it, right? You want to make sure you get the name right. So making sure that proofreading ensures more accuracy, right? That's a very, very important thing. DNA Polymerase III has proofreading activity. DNA Polymerase I has proofreading activity. Okay? Some DNA polymerases do not have proofreading activity. And these DNA polymerases end up being very, very much more prone, that is 100 to 1,000-fold more prone to making mutations than those that have a polymerase activity. One of the enzymes that is very mutation prone, HIV's DNA replication enzyme. I'll say more about it later. It has no proofreading activity. It makes an error, it blithely goes along its way and doesn't pay attention, just keeps making more. Okay? It's for this reason that HIV mutates very rapidly and is able to adapt to various drugs because it mutates its way around them. Now, for a cell, high rates of mutation are problematic because the more mutation that you have, the more likely you're going to form, in the case of our cells, a cancer. In the case of a virus, the virus, all it cares about is going on and making more viruses. If it mutates and it makes a non-functional virus, it still can make 1,000 more that are functional, and it goes along and does its thing, eventually figuring out a way around the individual drugs. Okay. Questions about that? How much of this is review? Some? Yeah, okay. All right. Let's see. What else do I want to say here? Lagging strand, leading strand. I've talked about single-strand binding protein. I've talked about ligase. I've talked about helicase. I've talked about topoisomerase. I talked about the beta clamp, and that's a up-close and personal view of the beta clamp. And as I said, the beta clamp actually gets loaded at the replication origin, in this case of E. coli. And the loading of the beta clamp involves taking something that looks like a C and basically closing the jaws of it. And in the process of doing this, the DNA polymerase is also attached to this. Okay, where are we at? Now, as if I didn't give you enough complication for the polymerase replication fork, I'll give you another one. Okay? This guy is odd. Okay? Leading strand synthesis, as you might expect, can occur more rapidly than lagging strand synthesis. Okay? Well, if the replication fork has to move, both of these have to be replicated at the same rate, what happens? Well, it means that the leading strand replication can proceed, and then this lagging strand, which hasn't been replicated, starts looping out. Like you see here. It starts looping out. This looping out process, okay, is called the trombone. Well, what happens? Does it just get bigger and bigger and bigger? No. Once the trombone gets to a certain size, leading strand synthesis slows down. The polymerase gets working on the other one. That is, working it back down. Okay? So the trombone allows the loop, allows the leading strand to continue for a while. It gets to a certain point, and then it stops. The leading strand synthesis stops, and the polymerase looks over here and does its thing. Now, when you look at the YouTube videos that I have out there, you'll see this trombone forming, and again, the orchestration of the proteins coming in and doing their thing within here. Okay. How am I doing here? Okay. What's that? Somebody said joke. I thought I heard joke. How about a song? Now, the song itself is a joke because the song has nothing to do with biochemistry, but I thought, given the fact that the weather is getting ready to change, we should have a song about the weather. Okay? So, some of you noticed I was whistling a song when I came in, and that's actually the subject of the song. You guys know the song Let It Snow? The Midwest knows the song Let It Snow. Well, in Oregon, we have our own song. It's called... All right. You want to join me? Oh, the Oregon weather's dowdy, 'cause the sky is mostly cloudy. You can't stop it if you complain. So let it rain, let it rain, let it rain. It doesn't show signs of slowing, and it's rarely right for snowing, though it's driving some folks insane. Let it rain, let it rain, let it rain. When it finally turns out dry, we'll be putting away our rain gear. It will probably be July, but I'll surely miss the reindeer, 'cause the sound of the falling rain, pitter-pattering down the drain, makes music inside my brain. So let it rain, let it rain, let it rain. Not too much enthusiasm, but that was there. It is getting ready to rain again. So anyway, I thought that'd be appropriate today. Let's see. How should I do this? What do you guys say we take an early day? Surprise, yeah. Hi, how you doing?
Would it be possible to take the next exam on Wednesday earlier?
Why?
I have like a family vacation thing that we do every year.
Well, family vacations, no. Generally, I would say no, because again, that's something that really, for professional development or some other problem or something, I could say yes, but I can't do it for that. Sorry.
All right.
Yeah. Sorry. And I do that with everybody, so it's not just you. Yes.
So in this scheme, the only thing that DNA Polymerase I does is replace the DNA or the RNA.
Replace the RNA. That's right. Uh-huh.
And DNA Polymerase III does the rest of it, right?
That's right. Yep. Hey, how's it going?
I was talking to Cody yesterday.
Yes.
Our class that we had.
Uh-huh.
He was telling me that it used to be like a news thing.
I still do it. Uh-huh. Yeah. So it's called News Group, and what we do is we get together at Bombs Away about once a week and sit around and talk about the news. You want to join us?
Yeah, like when is it?
Well, normally it's Wednesday. It's this term it's Wednesdays at 4, but these past 2 or 3 weeks I've been busy, so we're not going to do it probably until either the 23rd or the 2nd of March. I'm not sure yet, but send me an email. I'll put you on the list. You get notifications. Yeah. Hey, guy, how's it going?
Good, how are you?
Fine.
I just had one question on when I was reading the book the other day. This is from a couple weeks ago. But I was reading about the elongases and the desaturases. And they talked about them being embedded on the endoplasmic reticulum, but on the cytoplasm side. So that still takes place in the cytoplasm. It seems like on your highlights it said in the ER. Or yeah, that's just embedded on the Okay. So it's just, but it's still, so it's all something. Right, it is. I believe that. Okay. No, thanks. Take care.
I was going to ask you about the beta example again. I was thinking about that when you were explaining it.
Sure.